Hi, my name is Vitória. I am a graduate student at University of São Paulo, Brazil at Irene Yan's lab, and my project is entitled In Embryo and In Silico Mapping of Gene Regulatory Networks During Commitment of Sensory Neurons. So, the dorsal root ganglion is one of the components of the peripheral nervous system, and anatomically, he is located bilaterally to the spinal cord. So, here we can see both dorsal root ganglions bilaterally to the spinal cord. And in the interior of this structure will be located the cell bodies of the sensory neurons. So the sensory neurons can be divided mainly in three subtypes: the proprioceptive neurons, the mechanoceptive neurons, and the nociceptive neurons, and those neurons will respond to different stimuli. For example, proprioceptive neurons will respond to movement and position stimuli, the mechanoceptive to mechanical shocks, and the nociceptive to pain and temperature. So, as the focus of my work is to analyze the development of the dorsal root ganglion, the dorsal root ganglion is formed by the neurocrest cells. So, during neurulation, the neural tube and the neurocrest cells will be formed. So, here we can see the neural tube closed, and here those cells that are migrating are the neurocrest cells. So the neurocrest cells will give rise to different structures of the embryo body, and one of those structures are the dorsal root ganglions. So after the migration of the neurocrest cells to form the DRG, those cells will proliferate and differentiate. And in this way, the cells of the DRG will get organized in a manner where in the periphery, here in yellow, will be located the cells in proliferation, and in the interior, in the center of the DRG, in blue here, will be located the cells in differentiation or differentiated. So this organization can be observed through all the DRG development. So in the future, the differentiation of those cells into the subtypes of sensory neurons is dependent of the temporal expression of specific genes. So here we can see a resumed gene cascade, and this gene cascade is divided in a lot of different phases. So here we can see the phase of proliferation marked by the expression of PAX3, the phase of commitment marked by expression of NeuroG1 and NeuroG2, the early differentiation marked by the expression of islet1, POL4F1, and the early and late sensory lineage differentiation that will be marked by the expression of specific sensory lineage markers. So due to that, in each specific phase of this differentiation, we will have different marker genes. So the hypothesis of my work is that SCRATch2, that is a neurotranscription factor that is expressed in chicken DRG during development. So here we can see the expression of SCRATch2 restricted to the nervous system, so in the spinal cord and in the cranial ganglia. At this stage, we cannot see the DRGs yet. And as SCRATch2 is expressed during development in the chicken DRG, our hypothesis is that SCRATch2 can act during the DRG development. So he's expressed in this gene cascade and can act during the DRG development. So our first specific aim was to identify the position of SCRATch2 in this gene cascade of the DRG differentiation. So for that, we tried to identify genes that are co-expressed with SCRATch2 in the DRG. So through the analysis of single-cell RNA-seq data and through analysis of immunofluorescence and in situ hybridization. So with the single-cell RNA-seq data analysis, we analyzed published mouse embryonic DRG single-cell RNA-seq data, and through the analysis of the comparison of expression of SCRATch2 with the expression of other markers of the different phases of DRG development, we observed that SCRATch2 is not restricted to a specific sensory population in the DRG, and it is co-expressed with early differentiation markers. So here we can see that the clusters that have the higher expression of Scratch2 also have higher expression levels of islet1 and POL4F1 that are early differentiation markers. Next, to confirm our in silico data in the embryo, we analyze by immunofluorescence and in situ hybridization the expression pattern of specific markers, so markers of the specific phases of the DRG differentiation in chicken stages equivalent to the stages that we analyze the DRGs in the single-cell RNA-seq data. So here we can see that the neurocrest cell migration marker HNK1, the proliferation marker PAX3, the commitment marker NeuroG2, early differentiation markers, and the late sensor lineage differentiation marker. And what we can see here is that Scratch2 in both stages is co-expressed with early differentiation markers. So here in this central region of the DRG where will occur the differentiation. And if we observe the expression pattern of NeuroG2 and compare it with the expression pattern of early differentiation markers, we can see that there is a complementary expression pattern. So if we overlap NeuroG2 and Scratch2 or islet1, HUCD, or POL4FE1, we can see that there is this complementary expression pattern. And this can indicate that Scratch2 is not expressed during the commitment phase, but during the early differentiation phase. So next, we wanted to verify if Scratch2 could regulate the expression of other transcription factors present in the DRG gene cascade. So when transcription factors occupy the same hierarchical level in a gene cascade, possibly they can interact with each other, forming a gene regulatory network. So here I'm going to mention two types of interactions between transcription factors: the cross-regulation, where a transcription factor binds to a specific target site and regulates the expression of the transcription factor B, and vice versa, or the interaction of joint modulation, where a transcription factor A binds to a specific target site, the transcription factor B to another target site, and together both will regulate the expression of a specific transcription factor. So first, to verify the cross-regulation interaction with our data, we analyzed neurotube cut and run for Scratch2 in the chicken embryo and ATAC-seq in mouse to identify Scratch2 target sites in the genome. So we searched for intergenic and intronic regions near to islet1, POL4F1, or NeuroG2, and verify if Scratch2 could bind to those regions. And with that, we get two regions here in red that Scratch2 can bind, and near to islet1 or near to NeuroG2. So this can indicate a possible interaction of cross-regulation where Scratch2 binds to a specific target site that could be those regions and regulates the expression of islet1 or the expression of NeuroG2. So next, next we search for target sites that were enriched in those regions where Scratch2 binds near the transcription factors islet1, POL4F1, and NeuroG2, because when the regions are enriched for binding sites of other transcription factors, potentially those regions are regulatory regions. And with that, we observe that in those two regions where Scratch2 binds, here in red, those regions presented Scratch2 target sites and also target sites for POL4F1 or islet1. So this could indicate a possible interaction of joint modulation where Scratch2 binds to a specific target site and islet1, POL4F1, or POL4F1 or NeuroG2 could bind to another target site, and together both can regulate the expression of islet1 or NeuroG2. So to confirm in embryo the possible regulatory role of Scratch2 in the expression of islet1, We overexpressed Scratch2 in tick embryo DRGs and counted the number of islet 1 positive cells. So we can see that the DRG that overexpresses Scratch2 presented a higher number of islet 1 positive cells. So this indicates for us that Scratch2 activates the expression of islet 1 in the chicken DRG. So I would like to thank everyone in my lab here in Brazil, the University of São Paulo, our funding agencies, and our collaborators. And thanks, everyone.